Any other questions? And if no questions, we can switch to our next presenter. Thank you, Parag. It was very exciting presentation. Thanks again. Yeah. So I want to introduce Professor Reggie Edgerton, and Professor Edgerton is a pioneer in multiple fields. I will say it's maybe hard to count all those points. So definitely many aspects of muscle physiology, neurorehabilitation, task-specific rehabilitation, and many aspects of spinal cord stimulation. It's all that been growing in his hands, and it's really great excitement that he was able to join us today. More than that, I want to say that Dr. Edgerton was a great mentor for many of us, and it was always a great experience to work with him and hear his talks. So I'm not sure if let me check. So, Parag, do you know if Reggie is?
Yeah, he's on.
Yeah,
he's on.
I see. Awesome, fantastic.
Okay, I can't see anything. So.
Oh, we can see now. Yeah. We can see you, but we don't see your screen. Okay, now we see your screen with Zoom link to the conference.
Yeah. Okay. I'm trying. Okay.
Yep. Awesome.
All right. Okay. Thanks, Igor. It's great to have a chance to really a special situation here with so many people with interests that are so similar to ours. And given that I've only spent a few years on this, but it's all coming together and resulting in a lot of changes that I did not expect, and I don't think anybody else expect. Now, in my presentation today, it's going to be basically conceptual, and I'm not going to try to convince you of any particular results that we have obtained. But to me, one of the exciting things is how are all these organ systems responding so effectively with a very similar type of neuromodulation. Each one is a little bit different, and even with the same dysfunction and different individuals, you have to fine-tune the neuromodulation. But it is still, to me, remarkable of how these systems are responding exactly as that. That is a complete system. And what I have continuously found is that there's not very many people besides this particular group primarily that looks at this from I would describe as a top-down method, looking at the bigger picture and then trying to go deeper into the problem. So of course the key question is what could be the common mechanisms that could result in the improved function of all of these organ systems, spinal and supraspinal? And it has become quite clear relative to one of the previous questions that the spinal stimulation clearly is having supraspinal effects as well. And so that goes back to the same concept again, is the brain does not function in isolation, and neither does the spinal cord. It's the two together that is able to accomplish such what I would consider magnificent results, which seems that it is challenging. Let me make one more comment about the mechanisms. There is no one mechanism that's responsible for this. There are multiple mechanisms at multiple levels in the design of the central nervous system. So when someone tries to say, what is the mechanism of these effects? That is not a simple question. To have such a complex physiological response, there's no way that a single mechanism is going to explain this. And so that makes it more difficult, but also it requires us to open up our minds a little bit in terms of how the nervous system is functioning. Most people, the common response, I'm sure that you've heard as well as I have, is that many of the things that we see are impossible. And I think the right answer to that is it's impossible if you buy everything that you thought you knew from the beginning. But if you loosen up your mind a little bit, you may realize that there are other things that's going on. So I've been involved in developing the technologies that's necessary to get these things to general public. And now I'd like to start with in controlling movement. It really boils down to two things. I make this point over and over, and both of these points, I would say, are principles that are basically function automatically. So I like for you to keep thinking of the automaticity of how all of these things are happening. And one is the related to the how do we adjust the recruitment of motor neurons within a motor pool. And this is pretty well explained through the years of experiments that's basically led by Henneman and his colleagues. And so the important point is how does the system automatically decide which and how many motor neurons are going to be recorded at any millisecond during a movement? And so that one we have a pretty good idea on. But the next one is a long ways from having an explanation. And that is the importance of the pattern of activation of all motor pools. We can understand how we activate a motor pool, but the key is how do all of the motor pools that's involved in a movement, how do they get recruited? And the reason this is important is that defines, but it's very specifically what the movement's going to be. For example, it's been demonstrated decades ago that these spinal animals, even without any input from the brain, they can walk forward and backward and trot and so forth, and even step sideways. So, but this is an area which we do not understand, but there's a lot of interest in this, and I'll talk a little bit about that. So the first one, the size principle, I'm just quickly going to go through this. I'll make a couple of points. Number one, this represents the motor neurons that would be in the motor pool of most muscles. And the really exciting thing here is that the physiological and anatomical properties of the motor neuron matches amazingly well to the physiological properties of the muscle. Now here it shows these different colors which represent different properties of the muscle fibers, but the muscle fibers that are innervated by a motor neuron have relatively similar properties. They contract fast, they have certain metabolic properties. And the same is true of these other, another fast unit, fast fatigue resisted, and the slow motor units. So the final point is basically what drives that is how much current that goes to the motor pool. And this is why I want to emphasize here why we think, how we think to some degree, the animals and the humans can respond so well after complete paralysis. So here is, this represents the motor neurons of a motor pool. And the more current that goes into this motor pool, the more force, the more power. Most of the activity during the day is from these slow motor units, and you can modulate supraspinally, and proprioception can modulate. So, but the point is you're paralyzed if your input is less than what this motor threshold is. And as Parag pointed out and emphasized, most of our neuromodulation is below the motor threshold. And in the first experiment that we did with epidural stimulation, we found out that basically the results tell us that after complete spinal cord injury, that this might say resting potential is reduced. And so it could very well be, and we think what might happen is that after spinal cord injury, the excitability of these networks is decreased. And even if you have some supraspinal input or proprioception, then it's still not enough to get above the motor threshold, and the person's still paralyzed. So, so here is what if we elevate with neuromodulation, this resting potential, then we can get above this threshold with different sources of sensory input. So this basically takes care of, this addresses the issue of size principle. The next one is, how do we coordinate these motor pools? Now, all of you probably recognize this type of drawing from Cahal, but the point that I want to make is his drawing shows this, all of this input into this region of interneurons that then project to flexor and extensor motor pools. Now, the point is, what is happening here is really defining what coordination occurs, and therefore defines what the movement is going to be. So if we go here, for example, and look at which motor pools and motor neurons are going to be activated, it's determined primarily by the interneurons. And then with the emphasizing proprioception, as the proprioception changes, it activates different combinations of interneurons, and one combination of interneurons will activate one set of motor pools, and another combination will activate another. So these interneurons are used in multiple movements. It's not that each one is unique for a specific movement, which is something that seems to be an idea that's dominated. So I'm going to talk about proprioception. I'm going to talk about, these are the concepts that I think are important in what we're seeing in these conditions of different types of paralysis. How important proprioception is, the feed forwardness of it, Yuri Gerasimenko and others have published a paper on the importance of the feed forwardness. Often this is thought of as a phenomenon of the brain, but it's definitely a major feature, an important feature of the spinal networks. The other I'd like to address, and one way that helps to explain the observations that's seen with neuromodulation, is the redundancy. So, and some would refer to it as noise. The bottom line is, I'm pretty convinced that to refer to this variation as noise is probably minimizing certain features of the design of the nervous system. And this design results in there being a lot of redundancy in the neural networks, not only in the spinal cord, but also the brain. And then I'm not going to talk too much about the neuromodulatory mechanisms very much in terms of the result, but the implications of these to neuromodulation. And then just briefly a little bit about the automaticity. Now, where does the I like to suggest thinking of the automaticity is the result of all the input that's going eventually to the spinal cord. And of all these sensory systems, you can use different criteria for how important they are. Proprioception, I would say, for multiple reasons, and hopefully I can get to some of the major points, that proprioception is probably one of the major sources of input to the spinal cord, and in some ways even more important than cortical control. So now this particular experiment was one of the first ones that we were able to perform in the 80s with cats. And this is a follow-up of the experiments that have been done in Grillner's lab in the late 70s. But the bottom line here is this animal could not step. It could stand, but could not step. And it had been paralyzed for 3 months. And we only gave it strychnine intraperitoneal, and then 30 minutes later, we tested it on the treadmill, and it could walk over various speeds, full weight bearing. So we changed the physiological state of the spinal networks to a point where it could respond to proprioception. The only way you can explain this control, the source of control, is through proprioception. And this is an experiment, again, come from the Russian colleagues. Nessa Kozlovskaya was one of the originators of this from the space program. And Yuri has been using, Yuri Gerasimenko has been a key factor in using this and trying to understand the spinal control. And this is a very important experiment, I think, and I'll follow up. Okay, I won't go any further with that, but the point is, it's got force that's activated the heel toe, and you could hear the frequency. And just you ask the person, this is an uninjured person, you ask the person to try not to step. And then, but what most of them will do is their legs will start to walk and start to go into a rhythm similar to walking. All right, this goes to the next experiment that the idea came from Gerasimenko again, and this was experiments done at Veliki Luki, which is a fabulous laboratory that we've been able to collect a lot of great data. And then, so here's the situation: you ask the person to voluntarily step in a stepping-like motion, and then you stimulate, neuromodulate the lumbosacral cord, and you get these patterns of activation, these EMG patterns. So you say each motor pool has a unique pattern. Now we ask them to do the voluntary activity, and then we stimulate the sole of the foot like you just saw. And you get a pattern that is remarkably similar for each motor pool. This is remarkable, and it's telling us something very important in terms of the neural control. And it's remarkably similar to what has recently been reported in the brain. And basically, the bottom line in those experiments was no matter where, not no matter, but in receiving different modes of sensory information, they end up being activating very similar areas of the brain. So even though the different sources, the inputs coming from spinal stimulation is coming from the base of the foot, or you combine them, and all of it, by the time it gets to the spinal cord, it's all going to the same thing, and the spinal networks recognize this as stepping, and obviously generates the stepping pattern. So in the feed forward aspect of this, it continues to be increasingly impressive. And again, I remember being in the lab of Yuri Gerasimenko and Pavlov, not Pavlov, Pavel Muzizenko, where they did this experiment where they decorticated the animal, and these data have been published, but this was remarkable to me. If you look at the ground reaction forces of the right and left leg, you see how much they vary. but they're highly coordinated as they take multiple steps in a sequence. They're highly coordinated as you show here. Now, if we take those steps and we model this and randomize the order of this, the animal would not be able, the animal would fail to step and would fall down after about 5 cycles. Whereas if it's kept in the right sequence, it can go on for 15 or more cycles. So what this tells us is that whatever happens in one leg, it's already the network of the spinal cord has already told the nervous system, the spinal networks that's going to regulate the other leg has already had influence on what the gravitational force is going to be in that leg. In other words, the short story is whatever is happening in one leg, it's already preparing the other leg for what's going to happen next. And what's going to happen next is going to be one that's appropriate to maintain the balance. That, to me, is a remarkably important observation that they made. Now, going now on to the, how does redundancy and noise, how does this fit into the picture? And why is this important for us to think about in the mechanisms of control of movement? Well, here's an experiment that was done by Takiola in our labs several years ago. And this is an animal that intact rat, 20 days after implantation of electrode array. And what he did is stimulated from a specific area every 30 seconds. And look at the variation. This is the variation that's intrinsic to the spinal circuitry by getting the same stimulation. This tells us how important the other variable is and the output of the nervous system. It's not only the input, but the output is also determined by what the physiological state of the networks are. This is a feed-forward phenomenon that helps us to prepare for what movement's going to be. And here's the evidence. If you record from the tibialis anterior and from the soleus muscle, you see how they're coordinated, but they're not exactly the same. But more importantly, receiving the identical stimulation only one time every 30 seconds, and look at the variation. The only way you could consider where this variation comes from is from the constantly changing physiological state of the nervous system. So this is another area that actually reflects the feed forwardness of the spinal cord and reflects a very important source of control, which I won't have a chance to go into. But one more point I'd like to make. In this experiment, if you anesthetize the preparation, this is what you get, the red line. And this is what you get before the anesthesia. So this is a point of importance, because this difference between the networks here and the networks in an anesthetized preparation. You've eliminated maybe 90 to 95% of the nervous system in an anesthetized preparation, and therefore we should be very cautious in interpreting our experiments when in a very reduced preparation. Another reason we think that this is important is because we ask the question whether it might be important, this variation, how important it is for the spinal animal to learn how to step. And if we take the robotic device that's attached to the ankle, we train them, and the ones that were trained with a variable capability, which I will show in a minute, they learn to step much more regularly than the others. So, if, for example, if in the trained animals for a fixed trajectory, that is, the robot arm was always, it repetitively performed the same movement, as opposed to if we put a force field there, we gave them an opportunity. There was flexibility in exactly where the leg would go from 1 millisecond to the next millisecond. And also we controlled one leg versus the other leg with a force field. So anyway, in the end result, you get a lot of variability if you keep it a constant manipulation, as opposed to giving it a window of opportunity. And so this is what happens with the coordination. In the fixed trajectory, this is what happens when you look at the soleus being activated and the tibialis anterior in the fixed. In the assist as needed, as shown by the force field, you get a very clean difference. In other words, what if you look at the EMG, which I'm not even going to show, it's almost constant co-contraction here. And here, when you leave the option for the nervous system, the spinal networks, to make a decision of where the foot is going to go next, it can maintain a highly active and accurate locomotion. In a more recent experiment, going back to the variability, and as opposed to having very specific pathways for one motion versus another, this is the cross-section of the spinal cord. This is the density, as you see here, is the density of neurons that were activated as indicated by CFOS. And this is in a resting animal. And look at the amount of information going to the dorsal roots, going from the dorsal roots to the dorsal column relative to the intermediate and the ventral is quite amazing. But the point here is when we had these mice, that we could measure CFOS in 2 different ways. And so therefore we had the mice on one day to run a half an hour, and during that half an hour they took about 7,000 steps. And then a few days or a week later, we have them to repeat it. And what we found is the number of neurons, the number of primarily interneurons that were activated in both of these run-in sessions options were only 20%, only a 20% overlap of the neurons that were activated under both conditions. This goes toward the concept of redundancy. And one of the advantages potentially of the redundancy, it tells us how many ways there's possible to get from A to B. And so going on to the next idea of, and this fits in with the plasticity, if you have all of these options for reorganizing the networks, then that gives you a huge advantage in being able to find new connections and develop novel connections, which we know there's numerous experiments that's possible. So each of these things fit into the fact that the connectivity between the brain and within the brain and the spinal cord and between the spinal cord and the brain, there's tremendous amount of automaticity. There's not a whole lot of, quote, decision-making events that's really occurring. The nervous system is designed so that if a given sensory information occurs, that it works on a probability phenomenon. This is an individual that been paralyzed for several years, and after 5 sessions of working on a robotic device, we tested this individual to see if they regained voluntary movement, like some of the previous studies that subjects that we studied. And then we asked him to move his leg without the stimulation on, and then we asked him to move it with it on, and then after Yuri again gives the signal, and this this person had not been able to make a movement like that since he had been paralyzed several years earlier. So there's tremendous opportunity for plasticity if we can figure out how to take advantage of the automaticity and the way to activate the plasticity. And where I think we are right now, if you look at the general concept of the severity of a spinal cord injury and the functionality that you potentially can regain, it would be something like this until you get to a critical point where you're considered completely paralyzed. And basically, historically, these individuals have been told nothing can happen at this point. We think we're here. We think that that we can increase up to some level. We don't know what it is. We think also that we know so little about how this works that we're not even close to being able to identify how far up the scale we can go. But I think it's very clear that people with a complete injury does not have to assume that this is the amount of function that they're going to have for the rest of their life. Using these different techniques and understanding better on the mechanisms involved leaves us lots of potential. And I'd like to point out two individuals, particularly from my way of thinking when I started in this route, was Peter Zanger and the experiments that he did with Grillner in the mid-70s, showing that the complete spinal cat could regain stepping, and showed the concept of central pattern generation. And of course, that has continued in very robust way with Sten Grillner over the time. And so many people have contributed to this. I can't go into detail about any of them, but all of them are important. Yuri and I have such a fantastic time for years sharing our ideas, and Ruslan opening his lab up in Veliki Luka is fantastic. You heard from Parag, and then you know about Susan Harkema, which has got an amazing lab in Louisville, and Hoi Zong has been with me about 20 years now, and shows she's been very fundamental from Caltech, and Joel Burdick, and neuroengineer Wen-Tai Liu. So I'll stop there and ask and try to answer questions.
Thank you so much, Reggie. It's an extremely exciting talk and very insightful. Thank you a lot. It's I can feel a number of questions and answers expanded in both ways. May I ask you maybe a general question first? So since today we've been talking about different approaches like invasive, non-invasive neuromodulation for spinal cord, and you know all those data, of course, and the whole history of development of this field. So how do you feel about the right spot for either approach, and what will be the best next steps for those people who want to study spinal cord neuromodulation for spinal cord injury, to implement transdermal stimulation, epidural stimulation in combination with other techniques? Like, what do you think will be the best advice in this case? Because again, many people today expecting to start study or looking forward to move in this direction. So I think advice from you will be extremely helpful.
Well, I have a very clear answer here, and that is every one of these techniques that showed promise, the transcutaneous as well as the epidural, should be developed to its fullest. I'm convinced that there will be some subjects eventually that the logical choice would be epidural implant. For others, it would be transcutaneous. There's so many conditions. I think there's definitely more options with the transcutaneous, and it has advantages, like particularly with children. And rather than making a chronic implant in a child, I think to me, it makes sense to start at least with transcutaneous stimulation. I've been impressed with the efficacy of both of these. And I would only put my money on transcutaneous stimulation because I think it can eventually be used for more people more easily with less cost. But we haven't even mentioned, and we know that the other way to neuromodulate the system is pharmacologically. There is no question about that. And then we also know, I think, we've always obtained, even from the initial experiments, we always obtain more effective results when we combine stimulation and pharmacology. But this is another reason why I say we're just not, when I say we, the area, we're just beginning. We haven't begun to be able to identify the most effective way. But what this tells me is, given the design of the spinal cord and the brain, this tells me that there is a lot of potential here that we're not even close to having reached. So we should develop them all.
Thank you so much. Thanks a lot. Maybe I can ask.
Can I ask you, Igor?
Yeah, please, Igor Petrovich.
Reggie, thank you so much for your excellent, beautiful presentation. And also, I'd like to say thank you for possibility to work with you, Rich, almost 15 years. So it was, for my life, it was an excellent time. And so thank you. So my question about what the strategy for recovery, for rehab, what do you think? So when we stimulate, doesn't matter epidurally or transcutaneously, and for induce some movement, actually it is like neuroprosthesis. So, and at the same time, when we stimulate subthreshold, it is enabling. So what's the combination, or what's the balance between using the two strategy for inducing locomotion, or it doesn't matter, or staining, and so inducing and then trying to combine with the rehab, or it should be stimulation spinal cord for enabling, and then peripheral output during motor performance will be more effective for rehab the function which you practice. So what do you think, again, my question about, so the balance between the stimulation for prosthesis function versus stimulation for enabling?
This is a great question, Yuri. I think that we should not be thinking that we are neuromodulating, our neuromodulation is inducing the movement. There's, but I think you have, here's what I think, two things are happening here. One is, if you just train, you don't get very much plasticity in a complete. You get some, but it's difficult. So what you have to do is change the physiological state of these networks. And I think that's changing, it's making it more plastic. And this is what we don't know how it's doing it. But we've got to stimulate it enough to get it excited, to make it plastic. And I think that one of the things that is happening is we're making the networks we're putting it in a physiological state that's close to what I think of as the critical period during development. So when we, so this leads to, if we just stimulate and we do not rehab, that is, we have to show the sensory motor system, once it's plastic, we have to tell it how to reorganize. We can make it plastic and then it doesn't know what to do unless it's receiving some type of sensory motor activation, which I think the adaptation is driven clearly through activity dependent mechanisms. So the one stage is to activate it to a level so that you can begin to get these systems responsive. And then once you can get it responsive, then you can modulate it. You can strengthen certain combination of synapses. But I also think that that there's a tendency to overactivate. And when you, because what we're doing, we're trying to get to what's left in the spinal cord and brain after paralysis. We want it to work like it was designed to work. If we stimulate over a certain threshold, it's forcing it to work in a way that it has no choice. And we have to let it figure out what is the new solution. We have to let it relearn. And so I think, for example, we could train and train and train and get to the point where nothing happens because we're stimulating so much the system, we don't allow the system to reorganize as its intrinsic properties want to do it. And I think this is particularly important in the younger kids. And so we have to be very careful about imposing too much activation as opposed to gradually withdrawing our level of activation. As it becomes more functional, it needs less. That's theoretically how I think it works. But we have to definitely show that more definitively. But I'm pretty certain that you can interfere with the learning process if you push it too much.
Thank you so much, Rich. Yeah, I agree.
And Yuri, thank you for everything that you've taught me. And Igor.
And thank you both. Again, it was extremely exciting time working with you for last almost like 20 years. And Yuri for last maybe 25 years. So thank you a lot. Colleagues, please, if you have any questions, please ask.
Hello. Can you hear me?
Yeah. Maxim.
Yes, this is me. I've just noted. Well, actually, I have 9 questions. I think that I'm going to select most important at the moment, and possibly, if it's okay, I can just, I don't know, send the email if it's okay later on, for sure. What worried me in the speech of Yuri and later, you've mentioned that it's important, the importance of the patterns of the activations of motor neuronal pools, which again triggered my idea that there should be something much more complex rather than square pulses to activate the part of the nervous system, taking into account that the square pulses are not the language of the nervous system, and there should be much more complex pattern of activations of motor neuronal pools. So this is kind of comment and the question, isn't it?
Okay, this is a very good question, and
I think that we should not be thinking like what we're seeing is the result of a square pulse or any kind of shape of a pulse, even though I think that that has a factor. But think of it more as we're changing the properties of the neuronal networks. And I say neuronal networks because you can't explain anything that any of us are seeing based on one neuron. It's the result of networks of neurons. And we really don't have a good solid way to estimate the excitability of a network yet, like we have for a single motor neuron. But my point is, I think that when we stimulate, we're not, the response is not from a given pulse. The response is from what that pulse does to the membrane properties of the neural networks that makes it to respond differently. When you think of all the sensory systems, this is one thing that's always amazed me, is that we have these different modes of sensory input, but every one of them generally start, in large part, but not totally, start with an action potential. All the vocabulary is an action potential, but what the action potential does depends on what is the physiological state of the networks that's receiving those action potentials. So again, I would say if we just think of changing the membrane properties of the dendrites, the cell bodies, and the axons and the synapses, we're changing their physiological properties in some way so that then they're in a way that they can respond to supraspinal input that's normal and and the proprioception. I mean, it is, when you think about the impact of proprioception, to me, it's just flat out phenomenal. And there's the one slide that I show that I really couldn't explain fully the significance of it, but somehow proprioception and input from the brain, but somehow proprioception is activating a group of interneurons that can select the activation level of different motor pools. How does it know how to do that? My simple explanation is that the spinal cord has had over 300 million years to learn that language. And basically that's what, in my view, that's what's happening. That's clearly a hypothesis, but proprioception can do it. It can completely control what the leg is doing. And we've written a hypothesis paper that's related specifically to cerebral palsy. And I think the cerebral palsy is a good example of where we're using, what we're really trying to do is get the best proprioception going to the spinal cord, and that will help the spinal cord relearn how to move. And then that information can eventually get to the brain and cause plasticity, functional plasticity in the brain as well. So I would suggest let's try not to think about every response that we see from the nervous system is from an action potential. There is a lot happening to the nervous system that it doesn't even involve an action potential. This goes back to the invertebrate physiologists have known this for years, but I think we've kind of forgotten it. So again, think of it as modulating the physiological properties rather than inducing a specific response from a specific activation from an action potential. I hope you, let me apologize for the very short answer.
Oh, thank you very much. This is really important. And the second, I guess, important question is that you've mentioned that we have to create the model. And what do you think? What do you feel about that? Possibly the model could answer the questions that you have already said recently regarding the involvement and regarding the management of the proprioception and the influence of the proprioception over the selection of the set of the motor neuronal pools and the response of motor neuronal pools. Thank you.
Now you're talking about modeling of how you could model the spinal cord, the central pattern generator, the brain, how you can model that to generate the output that we see normally. Is that what you're asking?
Well, yes, my intention is like that. Yes, thank you.
I think one of the problems with that is you have to assume what the physiological properties of the networks are. And that's because I think almost all of the thinking is what the output is what we put in. And that is only, that's a part of the story, but it's by no means the whole story. So that's the problem with most of the modeling, is that this, quote, noise phenomenon, this is a real property of how our nervous system is designed. And I think there needs to be more attention paid to what's the physiological state of the networks that's going to receive the input that we've modeled.
Thank you. Thank you.
Thank you. This is a fantastic discussion. Reggie, may I ask you a question? Maybe I'm not sure if it's right way to ask, because you probably already give us answers, but still, going through major points you made in presentation, that spinal cord below the injury has whole capacity to activate multiple motor pools, right, and maintain same force and range of motion, and there is a great capacity to coordinate different pools and facilitate this coordination in many ways. So still, when we have to deal with spinal cord injury patients, again, with just a few fibers which left across injury, what will be a sacrifice when we're trying to focus on volume of movements, force versus precision coordination? And likely strategy could be different, depends on what you want to focus first, second, third, right, in rehabilitation process. So how do you see this process in a best possible scenario when it goes to clinic?
In general, if I understand what you're getting at, in general, I would say the coordination is by far the most important. In almost all of our movements, they require very little high forces. If you look at a number of studies have been done recording EMG on a 24-hour basis, and in fact we've published some of this from some of our monkey work when we were working with the monkeys in the space program, and John Hodgson basically reported, I don't remember the exact numbers, but about 80 or maybe 80 to 90% of the activity from the spinal cord to the muscles is at the very low range involving predominantly the slower units and the fast fatigue resistant units. Those higher, stronger, bigger motor units are recruited much less often. So anyway, the coordination, somehow you need to get enough force out of the motor pools that you can begin to get a somewhat of a normal proprioception that's going back to the motor pool. But I don't know what else to add there. But I would say just generally, I would focus on the coordination and the if you can get the right coordination, you've got enough force. You can't get it. You can't get the coordination without some critical level force. But what's really critical is the details of the coordination. It's not just it's not only flexor versus extensor. Slight changes in the order and the amplitude of the activation of a motor pool can totally change the movement.
Thank you. Thank you so much. It's very insightful. Thank you. So, colleagues, please, if anyone has questions. Anyone from the room or Carlos?
Yes. Thank you, Dr. Edgerton, for this beautiful presentation. You presented a beautiful timeline, and where you're talking about Sanger and Greenlaw experiments, I was thinking about CPG modeling. So, are we still talking about flexor emicenter, extensor emicenter proposes by Charrington and Brown more than 100 years ago, or there's something new on CPGs based on epidural stimulation and transcutaneous results? What do you think?
Well, number one, every time I go back and read what those guys did, I'm pretty impressed with their thinking. They didn't miss very much. Now, if you're getting to the point of central pattern generation, everyone thought, well, I don't know everyone, but most people thought that maybe we could get stepping with this neuromodulation type of approach. But what are you going to do when you get to the upper limbs, for example? And well, there may be some, but not very robust central pattern generation. Everything that we've observed and that has been observed in this area, the concept of central pattern generation is part of it, but it's not the central features of the nervous system that's important for anything I said today. In other words, I think the concentration on central pattern generation, which is where I started myself, was Grillner. I thought central pattern generation, that idea was pretty important, and I know it's important now, but not for the same reason. Most people have been thinking it is. And for example, most of the emphasis has been on it's important because it generates a rhythm it generates a rhythm and a pattern. And there's been all this controversy of which interneurons is responsible for the pattern and which interneurons are responsible for rhythm. And that may be interesting, but it's become less and less interesting to me with time. And because that's not why it's important. What's important for central pattern generation? I can't really say I've heard anybody say this, but I'll say it. the most important feature of the neurons that generate is that it recognizes the details of proprioception. In other words, the reason proprioception can be translated to the movement is because central pattern generation has already organized in itself of how and the sequence of what to activate next. So to get rhythm and to define the step periodicity, you don't need central pattern generation. Proprioception gives that to you. But the amazing thing is that the neural networks can recognize a specific sensory ensemble throughout a movement. And that sensory ensemble tells the spinal cord exactly which interneurons to activate, and that those interneurons know exactly which motor pools to activate to what a level. And that's what's been learned with 300 million years of evolution. So just stepping an animal, just the proprioception itself will give you rhythm and give you patterns. But it's much more than that. It's interpreted amazingly complex sensory ensembles into an output that's appropriate for what it's hearing from proprioception.
Thank you. Thank you so much, Dr. Edgerton.
Thank you. And maybe I can just follow this with this question. So, and that is likely because the number of facts and actually information about sensory processing is well, it's much limited, more limited compared to what you know about motor level, right? So I guess it kind of comes from both ends. So much more information was generated on motor side versus sensory input and in the system. And apparently more questions will come as we learn about how sensory input being processed across circuitry. And maybe it's again, will come to another level of circuitry questions.
Well, I'll agree with that. I mean, this is another way my brain has changed a lot is the sensory side hasn't been getting enough attention because the sensory side is telling the nervous system what to do.
Right.
I think if we could stop assuming that everything starts with the cortex, if we could open up our minds that far, we would be able to think more clearly, I think, about how the nervous system is designed to control movement. I would say, essentially, it's controlled by proprioception, not the details that's coming from the cortex. That's a large statement, but it's not one or the other. It's both in reality. Without we know, this is another thing that really changed my thinking. Parag came in one day and told me about this video of the guy that had lost proprioception. And I was amazed that the person, the people that have lost proprioception as an adult, they're basically paralyzed. And to me, the simple reason is it seems so logical, but we haven't been able to think like this. It seems so logical that that would be exactly the case, because your cell phone is smart enough to tell you what the answer is. And that is, if you want to figure out how to get to B, you have to tell it where A is. And if the brain does not know where any part of the limb, all these parts are, it can't tell you how to get there. So it can't plan, it can't do anything unless it knows where you are. Proprioception is central.